Hello everyone, welcome to another new tutorial of BTC 509 Bioinformatics Genomics. So in this tutorial I'll be showing you how you can extract an entire genome sequence FASTA format for your further uses. Okay, in order to search for a FASTA sequence you have to go to the server NCBI and say for an example we want to download the FASTA sequence of Oryza sativa. Okay, and for this case I'm pressing nucleotide as we want the nucleotide sequence for our case, and let's search for it. So Oryza sativa, I'm clicking it here again. Now you see this is the Oryza sativa japonica genomic sequence, and this is a complete sequence of chromosome 1. And now if I click FASTA, then you can see that this is a huge data file. Okay, you see this is downloading large sequence 1.4777, and it can be up to few megabytes. Okay, so you see this is a very large sequence and you can go up to down, and it is almost impossible for you to copy the entire sequence like the thing we did for our protein sequence analysis or amino acid sequence, right? Or even we can do it, we can just copy it from here in case of particular gene sequence as well, because that sequence is not that much big. But in case of entire genomic sequence or complete genomic sequence, it's really difficult to copy the entire thing. It will take hours for you to go to the bottom of this web page. So what we will do, there is an easy method to download the entire sequence, the entire FASTA sequence, which is you have to press here send to, and then you have to press file, and then you have to create file, and then it will download the entire FASTA sequence of your desired species. So you can see that it is still being downloaded in this page, and we are downloading our sequence, and if we click the sequence it will open up the sequence, and this is our sequence. Okay, one interesting fact of this FASTA file is we have .fasta extension after the file name. That means whenever we are having a PDB file we have something .pdb, whenever we are having Word file we have something .doc, and whenever we are having our FASTA file which has been downloaded from the website, it will contain the extension .fasta. Also one more thing, if you want to download a FASTA sequence of a virus, say for an example we want to download the FASTA sequence for coronavirus, COVID-19. Okay, so what we can do, we have a very specific server for this kind of virus FASTA sequences, which is NCBI and then virus. If you press NCBI virus, you can see we have a different server for this virus sequence, and you can go search by sequence or you can go search by virus. So we have the name of the virus, so we can type the virus name here, coronavirus. Okay, then it will show you different results, and for our case this is the one, severe acute respiratory syndrome coronavirus 2 SARS-CoV-2, which is COVID-19. Okay, so I'm searching for the sequences that are available. Also we have different filter here. Say for an example you want to get the idea about the sequences of coronavirus which are being extracted in the region of Asia. Okay, so you can write it there, and you can see we have different sequences here. Say for an example you want to download multiple FASTA sequences or multiple sequences for this kind of virus, and definitely you can have some all other informations like when this sequence was submitted, and then the name, and then the sequence length and the host, everything. Okay, so for now I'll be downloading two sequences of this COVID-19, say for the first one and for the second one. Okay, and then if you press download, and then as we want to download the nucleotide sequence, you have to press next, and then you have to press download selected records, the thing that we have selected. Okay, and press next, and then select FASTA definition line. You can use the default line name. Say at the top it will have its accession number, and then it will have its title. You can customize your title here, but we want to set it as default, and then press download, and it will be downloading your FASTA sequence. Okay, and it will contain, this file will definitely contain two sequences. One sequence will be the first one, and then the second sequence we will be seeing at the somewhere at the middle. I believe this is. So this is where the second sequence starts. Okay, so this is how we can extract our FASTA sequences for the complete genome. Okay, so that's it for this tutorial. In the future tutorial, I'll be showing you some important features of eukaryotic and prokaryotic genome.